This one? Okay, great. Okay, thank you so much for being here on this very warm Friday. My name is Erin Papke, and I am coming from the University of North Carolina, Wilmington, along with my colleague Liz, who we already talked about. She'll be giving a presentation on Sunday, and you can come diving with us on Sunday as well. And today I'm going to be talking to you about whether coral microbes can be friends or foes. So what is a microbe? A microbe is anything that is really too small to see with the naked eye. So you need something like a microscope to be able to visualize it. And these involve bacteria, viruses, some algae, along with fungi and other protozoans as well. So really anything that is just too small to see. And they're all around us. So microbes are really found everywhere. In any environment, they can survive a variety of temperatures and a variety of environments. They're at the depths of our ocean with hydrothermal vents. They're also in salt flats, they're in sulfur, they're in the Arctic ice. So even within the ice that is frozen, there are microbes in there. So they are all over the place. They're in the air that we breathe. We are covered in microbes, which sounds a little gross, I know, but they don't harm you. So they're everywhere. And they're even in the ocean. And the ocean has a lot of microbes, which can make it really difficult to study microbes specifically in the ocean, which is what our research mainly focuses on. So bacteria, for example, there can be about 1 million bacterial cells in just 1 milliliter of seawater, which is just a really, really, really small amount, and there can be 1 million cells in there. Now, there's even more viruses, about 10 to 100 times that many viruses in the seawater. So even more viruses than bacteria, and we're surrounded by it constantly. But microbes are essential for life. They are extremely important for every sort of system in every environment. They are responsible for breaking down any decomposing organisms. They're responsible for breaking down our food in our gut. So they're really integral to our gut health and our intestinal health. They help us break down all of our food. They help recycle nutrients back into the environment. So if we have a tree that's decaying, microbes will help break that down, or even an animal that might be decaying. And they're also really important for all forms of nutrient cycling. So there's even some microbes that can generate things like oxygen, which is obviously really important for us, but important for all forms of life. So they are integral, and they are all around us. Now, I'm mainly going to be talking about bacteria throughout my talk. And that's because our lab mainly focuses on bacteria and a little bit of viruses as well. But I'm mainly just going to be working on bacteria, talking about that. So there are multiple different kinds of bacteria, and this is kind of the friends or foes aspect of this talk. So we have commensal bacteria, and those are organisms that don't really harm you. So again, there are bacteria all around us. There's some that live on our skin, there's some that in our gut, and they're not really doing anything. They're not harming us. So they're really just commensal organisms. They're kind of just there. So they have their own processes, but they're not harming us, which is great. And we also have mutualistic microbes. Specifically, there's a microbe that they're required to put in some yogurts. So how many of you eat yogurt? Probably a lot, yeah. I eat yogurt probably every day. And yogurt has beneficial microbes in it, and that helps with your digestion. It helps break down your food, and there are some required bacteria that have to be put into yogurt to be considered like a healthy probiotic yogurt. Now, we also have pathogenic organisms, and those are the ones that will make us sick. They can cause disease, or they can cause any sort of harm. For example, Mycobacterium tuberculosis is the causative agent of tuberculosis. So in general, they will name the diseases after the organism that causes them, or vice versa. So it causes tuberculosis. This used to be a really, really impactful disease before antibiotics were used. It was also called consumption. So if you ever are really into history, consumption used to kill a lot of people. So it was responsible for a lot of deaths, but luckily now we have antibiotics, so not that many people get tuberculosis. And we also have TB tests, which you generally have to take if you're traveling out of the country or working with animals. So there are a lot of preventative methods to avoid these pathogenic bacteria. So again, we have multiple different kinds of bacteria. And just to break that down even more, we have the microbiome. So the microbiome is a generalized term for any of those microorganisms, but again, mainly just talking about bacteria here. So we have the commensal, as I said, where your microbe's pretty happy, your host is largely unaffected. And then we have pathogenic, where your microbe is really happy, but your host, like us, might be really sad. We might be really sick. We might be heavily impacted. And then we have mutualistic or our beneficial bacteria, where our microbe is really happy and our host is also really happy. So like that great gut bacteria that helps us digest our food. Now, this is a Venn diagram, so all of these can overlap. And this gets pretty complicated whenever you're talking about disease or disease dynamics, because a lot of microbes can actually switch from pathogenic to commensal, vice versa, or even commensal to mutualistic. It all just kind of depends on the environment and the situation, which I'll get more into. So in this in-between right here of our Venn diagram, these all overlap, and it gets really complicated. But what happens whenever these aren't overlapping or we're in a homeostasis, which everything is pretty much staying the same? Now that's called symbiosis, which I actually saw is down by our harbor, which I noticed today. They have like a nice symbiosis where everything lives in symbiosis with each other. So we are all currently existing in symbiosis. No one is screaming, no one is yelling, no one's getting upset. We are all happy and existing here, probably sweating a little bit, but that's okay. So symbiosis is just a close long-term association between two things. Generally, whenever we talk about symbiosis, it will be in terms of species. So for example, in terms of me and a dog. I love dogs. I live in symbiosis with the dog. If you have a pet, you're probably living in symbiosis with your pet. They give you happy joy, and you give them food and shelter. That's a type of symbiotic relationship. So we have multiple different kinds of symbiotic relationships, and you're probably familiar with some of them and maybe just didn't put it into this context. So for example, we also have mutualism, which the one species, one host is really happy, and the other species is also really happy. So a great marine example of this is clownfish and the anemone. The anemone provides the clownfish with protection, while the clownfish provides the anemone with some food. It kind of eats the things off the skin of the fish. And now we also have commensalism. So this is an example of a barnacle, which is hitching a ride on a whale. So the whale is really unaffected. It doesn't mind that there are a lot of barnacles on it, but the barnacle loves it because it can get a ride throughout the ocean, and it can filter feed everywhere that the whale goes, which is great for it. Now we also have parasitism. So there are a little bit different terminology between pathogenic and parasitic. So it kind of depends, but parasitic is generally for species impacts. So this example is actually a parasitic castrator. So this parasite will take over the reproductive system of this crab and castrate it. So this crab now thinks that this parasite is its baby and it wants to help it. It affects its brain function. So some parasites are really wild, and Liz studied parasites, so if you have more questions about parasites, she loves them and loves to talk about them. I find them like very terrifying. But that one is really cool. It's called like a zombie parasite because it takes over their brain function and impacts their whole system, which is pretty wild. And another example of symbiosis, which is probably why you are all here today, is coral. We love our coral. So corals live in symbiosis with a variety of different things, and I'm sure you're familiar with their photosynthetic algae that live within their tissues. This is commonly called their zooxanthellae. So this is what gives the corals their color. Without these, corals will generally appear white, which is why we have coral bleaching or that terminology. So the corals have to balance their photosynthetic dinoflagellates, and they also have to balance their microbiome. So again, corals have beneficial, pathogenic, and commensal microbes living within their surface mucus layer and also within their tissues. And there are also microbes even within the skeleton because, again, they're really everywhere. They're all around us. So the coral microbiome consists of all forms of microbes. They have viruses, they have bacteria, they have algae besides their zooxanthellae. They also have protists and archaea. So really everything exists in this coral microbiome. Now, together, these three aspects, because without each of these, the coral would not survive, is termed the coral holobiont. Now, this was a term that was created in about 2002, so a little bit newer of a concept, but the idea behind it is that the coral would not exist without its microbiome, and it would not exist without its dinoflagellate zooxanthellae. Now, because there are multiple different aspects, great, okay. Because there are multiple different aspects that associate with the corals have a really hard job of balancing their microbiome and their zooxanthellae. So we love to call this the balancing act, where we hope the scales are pretty equal, but whenever you have a disruption of this symbiosis, so this balancing act is toppled over to one side, that's a breakdown of the symbiotic aspect. And I'm sure a lot of you are really familiar with the one breakdown of symbiosis, coral bleaching, and that's whenever the symbiotic relationship between the coral itself and its zooxanthellae breaks down. So if the coral gets stressed through temperature or through ocean acidification or even sediment, or sometimes even if they just feel like it, they will expel, so completely release their zooxanthellae, and that's what makes the coral white. So the coral's not actually dead at that point. It just loses its color because the endosymbionts and the zooxanthellae are what give it its color. But if the coral exists for a long period of time without its symbionts, it can actually die. So it does take a while, and it depends on the coral species, but they do have the ability, if the environment goes back to normal, they have the ability to reuptake these symbionts and be okay. So they can reestablish this symbiosis if need be. They also have the hard task of juggling their microbiome. So again, all of this balancing aspect, trying to figure out how to not tip the scales. And this gets even more complicated whenever we're talking about a changing environment, such as our environment that is changing due to climate change. So you might be wondering, if corals have pathogenic microbes, and they have all of these microbes all around them, why doesn't every coral get disease? Why doesn't every coral have tissue loss? Why isn't every coral bleaching? Why is this not happening? Why is this not more common? And disease can be really complicated, so we like to think of it in terms of the disease triad. So whenever we're thinking about disease, we have to think about the host, which would be the coral in this case, the pathogen, which might be a pathogenic bacteria, and then also the environment. Now, each of these aspects has a variety of different factors that are associated with it. So for host, you can think about something like an immune response. I mean, if you think about it in terms of humans, one person might get really sick a lot and one person might not get sick that much. And that can be due to differences in your immune response. So corals also have differences in their immune response, and it varies based off of coral colony and coral species. So it really kind of just depends. But you have to factor in the host and all of these factors when you're thinking about disease. And then whenever you're thinking about the pathogen, there are also all of these other factors. See how I'm building on disease being really complex? So whenever we think about the pathogen, for example, if you think about the dose, if a stranger sneezes on you, you might not be impacted by it. You might not get their cold. However, if you're living in the same house with a spouse and they're sick, you might be exposed to their sickness more frequently, so your dosage of that pathogen would be higher. So that can impact this disease dynamic as well. And then also the environment. And I'm sure you're all familiar with climate change as we are experiencing right now with a lot of these hurricanes and it being very hot outside. So the environment also impacts disease in general, and also specifically coral disease in the ocean. So if you have high UV, high sedimentation, those are just extra stressors. I like to think of it as maybe if you're traveling a lot and your immune system starts to be impacted because of your traveling, you're exposed to a lot of different pathogens, you might be more likely to get sick. So the same thing can be said for corals. If they are in a really stressful environment, maybe there's a lot of sedimentation, their immune response might decrease, and then they can be heavily impacted by a pathogen. So again, we have this microbiome, and now I'm specifically going to talk about the pathogenic side, which would be in relation to corals. So specifically for bacterial pathogens, there's a lot of different coral diseases, but there's actually only about 12 known coral pathogens that are of bacterial nature. So not a lot if you think about it. It can be really hard to study coral disease on top of having a different colony response. There's different species that might be impacted, and different areas like the Caribbean or the Pacific or the Red Sea. So there's a lot of different factors that go along with this. But some known coral pathogens that we have identified as causative agents of these diseases include some of the ones listed here. So for example, black band disease, there are multiple known microbes that contribute to this black band. And black band disease is found within the Caribbean and the Pacific and the Red Sea, so it's really widespread. I didn't see any on these reefs, so I didn't see any disease on these reefs, which I only went to 2 sites, so don't quote me. But black band has multiple microbes that attribute to that black mat that sort of forms and then creates tissue loss. Another example is the causative agent of white pox, Serratia marcescens, which I'll actually talk about in the next slide. But that's an example of the elkhorn coral with white pox, so that's what it looks like. We also have the known agent of white plague type 2. There are multiple different kinds of white plague. They're all just named white plague in general. And then we also have some tissue loss and bleaching, which is Vibrio corlyticus, and that's a lot of what our lab focuses on, so I'll talk a little bit more about that pathogen as well. So Serratia marcescens is pretty interesting backstory. It's actually found in intestinal tracts. So we have Serratia marcescens in our gut along with other organisms, and it is generally found in sewage water. And if you ever get a little bit lazy in your cleaning habits, you might see something pink start growing in your showers. That is Serratia marcescens. So just a tip, maybe clean your showers a little bit more frequently. So it is found in our water along with our intestinal tracts. However, we think that It actually got into the ocean through wastewater runoff. So wastewater can be a huge contributor of coral disease and just releasing a lot of bacteria into the ocean. And some human bacteria can actually survive in an ocean environment. So this bacteria, which again is the causative agent of white pox, is a wastewater bacteria from humans, which is scary. And it was first found in the Florida Keys in about the 1990s or so. It only impacts elkhorn coral, Acropora palmata. However, it has reduced or did reduce about 50 to 80% of elkhorn coral. So a lot of the acroporids, which are the two different kinds of acroporids that we have in the Caribbean, kind of the third, have really been impacted by disease in the Caribbean. So that's why we don't really see them very frequently. And this reduced their population by a lot in the early,
I'm sorry, is the sewage water the only source of corals getting microbes, or is there other options too?
So Serratia marcescens is the only source of white pox for elkhorn coral. Yeah. Yeah, it's a little gross if you think about it. It's like our waste is causing coral disease.
Because we definitely have that here.
Yes. Yep. So it can proliferate in the seawater, so it can spread. Serratia marcescens can exist in a salty ocean environment as well, which is why it is the causative agent. So a lot of it can be really disturbing, and a lot of diseases have come from Florida, go United States, and a lot of like the Florida Keys and Miami, and that's because they do have a lot of runoff, and especially we're probably going to see a lot more impacts with the continuation of hurricanes, because that's really going to impact our wastewater treatment and where our wastewater might end up in the end. So just something to be aware of, and just like a not fun fact, but a fun fact. Now we also have Vibrio corlyticus, which again is mainly what our lab studies, and Liz will be talking more about in her presentation. So Vibrio corlyticus is widely spread. Widely found. We can find it across all oceans. So it's not just isolated to the Caribbean. It's not found only on Caribbean corals. It does infect corals, but it also infects other invertebrates, so things like oysters or fish. It heavily impacts oyster larvae and can cause a lot of mortality in oyster larvae, so it's really difficult for oyster hatcheries and things like that. And so we also know that it can cause bleaching. However, it actually induces rapid tissue loss, which is a little bit more disturbing. So here we have a picture of an acroporid coral that is from the Pacific, and about 24 hours later, the coral has almost completely lost its tissue. So this coral was dead by now, but it will die without any of its tissue. Obviously, it would be like you losing an arm and a leg and another arm. So this is really scary because it can cause this really rapid tissue loss, and we have no specific way of treating this type of infection, and it blows through corals so fast that we probably wouldn't even know it's there. It's just the coral is healthy 1 minute and dead the next. So this is also found in stony coral tissue loss disease, or SCTLD. Now this is a disease that our lab mainly focuses on and what most of my research is about. So stony coral tissue loss disease is an infectious waterborne disease. So we know it can transmit throughout the water column, and it can also transmit by direct contact. If corals are right next to each other, they can transfer this disease. And it has an extremely high mortality rate, so if a coral shows signs of SCTLD, it can actually die within weeks to months. And it depends on the size of the colony, but weeks is a very short period of time. It's probably not, depending on your monitoring and how frequently you monitor, this coral might be dead and no one knew what happened to it. So very fast it can blow through these corals. and it has an extremely large distribution and duration. It was found off the coast of Miami in 2014. So it's been 10 years that we've known that this disease has existed, and it has now officially reached about 30 countries and territories throughout the Caribbean. And I know it reached Saba in 2021. Again, I didn't see any signs of it out on these reefs, so that's exciting. But it tends to blow through reefs, and then it slows down a little bit because it went through all of the corals that it could kill, it did kill. So it kind of blew through, which tends to happen frequently. And the scary thing about this disease is that we don't actually know what's causing it. We have no idea. We think that pathogenic bacteria are involved, and Vibrio coriolyticus can actually cause tissue loss to be faster in this disease. So it causes the coral to lose its tissue even faster if coriolyticus is found. However, it's not the cause of this disease. So this disease is something that I've been researching and really trying to narrow down what the pathogen is and trying to figure out the best way that we can manage its disease impacts because it's decimating corals. It hasn't made its way to the Pacific yet, but we are a little bit afraid of that because we aren't sure what the impacts might be. And because of all of these reasons, it's actually considered one of the most devastating coral diseases ever recorded in history. So it is even like the COVID-19 pandemic of corals. So it's terrible and has completely decimated things. Okay, so that was doom and gloom, but now I'm going to talk about the beneficial side, which is the happy part for the rest of the talk. So beneficial bacteria are termed probiotics, and probiotics are live microorganisms. Whenever they are given to a host, they have some sort of health benefit. Now, humans have been using probiotics for centuries, and they're frequently found, again, in your yogurt that you eat, but also in things like cheese. And there was a huge probiotic phase like 10 plus years ago where they had like the Activia commercials and everyone was really into the probiotics. But it is really helpful for your gut health. So if you take a probiotic, a lot of people take them as supplements. So it's becoming more and more popular. So just to kind of explain the difference between probiotics and antibiotics, I'm sure you're all kind of familiar with antibiotics. You've probably had to take them at some point in your life. But probiotics are specifically in terms of coral. They're naturally found on the coral. So whenever we find our probiotics, we actually isolate them from coral themselves, so they're already there. But antibiotics are substances that are produced by microbes. So they might not be naturally found on the coral, and we are actually introducing chemical compounds to these corals that aren't naturally found in their environment, and we aren't sure what those impacts might be. Probiotics can also provide lasting protection. So the cool thing about probiotics is because they're isolated from corals and naturally found on them, if we give a probiotic to a coral, they can actually integrate it into its normal microbiome. So they can integrate it into all of the other microbes that exist on that coral, which is really great. But antibiotics are really short-lived. I'm sure a lot of you have taken a 10-day regimen of antibiotics. So after that 10 days, the antibiotic is not going to help you once you stop taking it. Probiotics can also be a preventative treatment, and this is called a prophylactic. So we can actually pre-treat our corals with these probiotics, and it can have different health benefits. But antibiotics are not preventative. You only take antibiotics after you've already gotten sick. And generally, just as a fun side, probiotics are a lot cheaper to produce, but antibiotics can be expensive because, again, we're isolating those chemical compounds. So I know this figure is a lot, but I just wanted you all to understand that probiotics can have a variety of functions. So for example, some probiotics can actually produce different vitamins. Specifically, probiotics for our gut and our gut bacterium produce vitamin K. So we can't actually produce vitamin K, but we do need it to survive. So probiotics can produce fun things like vitamins. They can also exclude and outcompete pathogens. So if you have a probiotic and a pathogen growing together, the probiotic might actually kill off the pathogen, and that's really great because we don't want our corals to get sick. So we have tested our probiotics on a variety of different things for a variety of different aspects. So we have corals that were at Nova Southeastern, and they contacted us, and they rescued these corals from, it might have been a barge or something where they had these corals in the wild. They brought them back into a land-based nursery, and they realized that their corals were really unhappy. They were having tissue loss. They weren't eating well. They generally just really didn't like that environment. So they contacted us and asked us if we could help them and send them some probiotics. So we actually sent them some of our probiotics, and after about 5 or so months, these corals recovered. They started growing back the tissue that they lost, and they were also feeding really well. So they actually succeeded keeping them alive in these land-based aquaria, and they were doing really well in 2021. We also have a probiotic that can directly treat stony coral tissue loss disease. So the current treatment for SCTLD is an antibiotic paste. However, the antibiotic paste can be extremely expensive. You need to reapply it to the corals, so you have to have a lot of boats, a lot of divers, a significant amount of time and money to be able to treat these colonies, and we aren't sure what the environmental impact of these antibiotics are. Since these corals are surrounded by other things in the ocean, we have no idea what the antibiotics are doing to the fish, to the coral next to it, to the algae, or just releasing antibiotics in the ocean may or may not be the best idea. So our lab is specifically looking at probiotics, which again are beneficial microorganisms isolated from corals themselves. And whenever we look at SCTLD, we have SCTLD that wipes its way through the coral tissue. So this is about 8 days, and the coral on the right doesn't have any tissue left. I know it looks like it does, but it actually got stuck in its surface mucus layer because it lost its tissue so rapidly. It actually just got stuck there, and it didn't slough off properly. But whenever we treat our corals with our probiotic, we can actually stop or slow the disease. So this can be directly used to treat stony coral tissue loss disease. Now, the really cool thing about our probiotics is that we can actually protect healthy corals. So this is something that antibiotics can't do, but our probiotics can protect healthy corals from transmission. So again, if we have a diseased coral, SCTLD is transmissible through direct contact or through the seawater. So if we put a healthy coral in contact with a diseased coral, the healthy coral will then be diseased, which is what is expected. However, if we put a healthy coral that was pre-treated with our probiotic in contact with a diseased coral, our healthy coral will not get diseased. So we protected our healthy coral. And the really cool thing is that because they're in direct contact with each other, about half of the corals actually gave this probiotic to this diseased coral, and the diseased coral actually stopped tissue loss. Yeah.
Are they the same genotype?
They are the same genotype, yeah. So that was really exciting that we can actually treat, yes, yeah?
Can you give us a simple explanation of what the probiotic is?
The probiotic is just a bacterial strain that we isolated from a healthy Montastria cavernosa coral, the great star coral. So what actually we noticed is that there were corals that got a lot of disease, and it was actually isolated in the lab. So there were corals that got disease, and this one coral just refused to have tissue loss. It didn't want to die. It didn't want to get STLD. It was really great. So my advisor actually was wondering, what is going on with this coral? Is it its genetics? Is it just being difficult? So he actually isolated MCH17, which is just a bacterial strain, and it actually produces a few antimicrobial compounds. And we're still kind of working through establishing what exactly it's doing and why it's so great at treating SCTLD. But he did publish a paper last year which has a lot more chemical compound information about this specific bacteria. Yeah.
Does it work against different strains of SCTLD?
So we have, yes. So does it work on different strains of SCTLD? Are you talking about like different species?
No, like when they mutate over time.
Okay, so we have tested it on a variety of corals, and we still are testing it. So it was isolated in, I think, 2017 or 2018, and we are still using it, and it still helps. So if whatever the pathogen of SCTLD is, if it has changed over time, it does tend to help Montassia cavernosa more because it was originally isolated from a Montassia cavernosa. However, it does help other corals, such as like an Orbicella or something, which they're slightly more genetically related. But the idea is that we are actually expanding our research, and we hope to find and isolate probiotics for a variety of different species and create more of a cocktail. So I'll talk about that more later. But the last thing from this slide is that we actually haven't seen any negative impacts of adding this bacteria to our coral. So that's really great. This bacteria is not causing any bad things. So we have been using this in the field. This is the first ever probiotic treated in the field. And we have two different methods. So this method is showing a colony treated method. And so basically we have a weighted rope and a plastic bag, and we will pump the plastic bag full of this beneficial bacteria, and then we will leave the bag on the coral for about 2 hours, and that allows the bacteria to be integrated into this coral microbiome. So this is a whole colony treatment, especially if you have a really big colony that you want to protect all of its tissue. This is a really great method. And we also have a disease lesion specific method. So what we can do is put this paste right on the lesion front, and this paste has the bacteria, the probiotic in it, and we can put it right on the lesion. And this way, the probiotic is in direct contact with the lesion, but it's still integrated into the coral microbiome. And this will stay on the coral for about 6 hours, and then it will just naturally dissolve. And it's a slightly different mixture from the paste that they use for the antibiotic treatment, and ours is not proprietary, so if you're interested in this, my advisor can give you the recipe. We don't charge for it, which is really nice. So our lab is based in North Carolina, which is up there. However, we have been working with a variety of different locations for our probiotic development. So we have this really great technology. We have a lot of robots in our lab which help us isolate these probiotics. And we can test these probiotics for a variety of different things. So antibacterial activity, whether or not they test well against some of our isolated pathogens. So there's a variety of different things that we test up in our lab. But basically what people do is just collect coral mucus from a healthy coral. The best thing is if you see a bunch of disease around, but one coral looks great, you want to test that coral, because something about that coral is helping it or protecting it in some way. So, yes.
Is there a reason why you're working in the British Virgin Islands but not the U.S.
There's no reason why we're working in the British Virgin Islands and not the U.S. Virgin Islands. They actually specifically invited us in 2022. Blake and I went to Grand Cayman, where they had a workshop on SCTLD specifically, and so that's why we actually tend to work with a lot of the British Virgin Islands or British territories like the Cayman Islands or Montserrat. We built a lot of those connections. But the U.S. Virgin Islands are mainly doing the antibiotic treatment. So maybe we would love to expand our area of interest. And specifically in Montserrat, I actually went to Montserrat last year, and we are working with Andrew and Emmy from Island Solutions. Montserrat's only about 2 islands away from here. And what we did is that we actually built a coral facility from the ground up. So whenever we got there, they actually only had the plot of land. They didn't even have these rocks or anything. And we're right on the water, and it was a beautiful location. And we actually built an entire probiotic testing facility. So this facility has an insulated water table, which is in the middle, and that's where we house our corals. And it also has a controlled water heating and cooling system. So we make sure our corals are not cooked and they're also not freezing. We get our seawater actually right off the edge of the land over here, and we filter our seawater to remove any bacteria or any contaminants. And we also have things like shade cloth, and they're running on solar panels and backup batteries. So we actually built this facility in about a week. Yeah, it was really great. We had working nonstop every day. We actually designed this facility and assembled it in Florida, and then shipped every component down in a cargo container, and then reassembled it in Montserrat. So that's how we built this facility. And actually, they've done a few initial probiotic trials, and they were just contacting me today about doing some field trials with those colony bags. So we've officially moved to the field aspect of this research. And this is really great because it can also be used just to house coral. So this can also be used for a restoration aspect besides just testing probiotics. So I think that they are interested in expanding this in the future. And this kind of is just proof that really anything is possible on a small island, even Saba or Montserrat, which is really great. And it is the first probiotic testing facility in the entire Caribbean. So because it's only 2 islands away, if you're interested, maybe we can set you up with Island Solutions and test some corals there. So the last thing I really want to talk about is that probiotics can be used for any life stage. So yes, we use probiotics to help with the health of adult corals, and we also use them to help treat disease. However, we are also using them for coral settlement. So we might use a probiotic on a settlement tile, and that encourages the coral to actually settle on that tile. We also use some probiotics to help with the initial stages of coral growth, like coral larvae. Once they initially settle, we're looking more at recruits, and we can see if our probiotics are helping the recruits grow or grow faster. So there are a lot of different things that we can do with probiotics throughout the coral's life cycle, and it doesn't necessarily have to be related to disease. So the main question is, can coral microbes be friends or foes? And the very scientific answer is that it just depends. So they can be friends or foes, but the main component that I want you all to take away from this talk is that they really are extremely important for the coral holobiont, but also the whole ecosystem and really just everything in general. They're important for food webs, and for all nutrient cycling. So they're really, really integral for the health of the entire globe. And this work wouldn't be possible without everyone in our lab, and specifically our advisor, Dr. Blake Ushijima. And we have many people who are working on this project. Our research technician, Margaret, she does a lot of troubleshooting with our robots and does a lot of the probiotic finding and isolation. So a lot of this work is actually stuff that she has done. And we have a lot of funders. Specifically, Island Solutions in Montserrat is funded by the Darwin Initiative, so they got these grants to build this coral facility, and that has really helped us help them, actually. But we have a lot of different aspects, and we have a lot of small funders and private funders that have been really interested in our probiotic development. And if you're interested, my contact information is up there. Also, if you just Google my first and last name, you'll find my website, and my contact information is on my website. And the large QR code will take you to our lab website if you're interested in learning more about our entire lab's research. Yes.
When coral bleaches,
Mm-hmm.
when it expels the
Zooxanthellae.
yes.
Yes.
What benefit is doing that to the coral?
Yes. So the question is, whenever a coral bleaches and it expels its zooxanthellae, how does that benefit the coral? And it's a little bit, it's not super well understood, but the hypothesis is that whenever the coral expels its zooxanthellae, it's because the zooxanthellae are actually causing them too much stress. And that's because the zooxanthellae are photosynthesizing too much. So whenever you photosynthesize any plants or anything, they'll photosynthesize a little bit too much, and they produce things called reactive oxygen species. And reactive oxygen species will cause cell damage. They'll bind with other things. So that is the main hypothesis as to why corals will expel them, is that they're too much reactive oxygen species are causing coral cell damage.
Well, I guess here we go.
Keeping on the theme of zooxanthellae,
Yeah.
I've heard rumors that they're investigating links between Skittle D and zooxanthellae because
Yes.
it seems to slow down when corals are bleached.
Yes. So I guess the question was, is there a link between the coral zooxanthellae and SCTLD, the tissue loss disease? And people are investigating that. That's actually what I'm investigating as well. So one of the hypotheses is that there are actually viruses that are impacting the zooxanthellae and somehow stressing out the coral and causing tissue loss. So I use a very highly specialized form of microscopy that allows me to visualize viruses. This microscopy can visualize things like mitochondria or cell walls. So it's very, very high magnification. There have also been different studies where people have done some sequencing to figure out if it's a specific kind of zooxanthellae. So there are multiple different genera. And so we aren't really sure. Again, it's a little bit complicated. We aren't sure what is actually causing this tissue loss. But it is one of the running hypotheses. However, depending on which research group you ask, people might say, no, that's not true, or yes, it is true. So it really just depends. But I'm hoping to maybe have an answer for you next year when I publish my dissertation.
Okay, if there's no more questions, then another round of applause for Erin. And we'd love to have you back when you can tell us more. And it sounds like probiotics may be a really cool answer to our Skittle D
Hopefully.
issues. Please make sure you pay your bills, everybody. Be nice to the venue. Thank you very much for coming. If you want to sign up for Sunday's Dive, see me or Emily. If you want to buy raffle tickets, we can do that. And if you're sitting on a white chair and can carry that just over to where we store our chairs next door, that's super helpful for the restaurant to be ready to serve dinner. So thank you very much, and we'll hope to see you on Sunday, diving and at the presentation. Thank you.